Hello, this video is going to talk about the serial dilution and what that all means, and then relate that to how the spectrophotometer kind of works and what this transmittance absorbent kind of stuff is all about. So, these are the pictures from your lab at the very beginning, and at the very beginning we made those serial dilutions. So we had all of the green stuff, I'm representing that by the red dots, that's what your picture had. And all those red dots, and as time went on we had fewer and fewer of a green color in our test tube, and I want to talk about why that is. So we'll start with the stock solution. That's a solution I gave you. Now if we look at this right here, it actually has 100 little red dots in it. So it's like it's 100%. So we can also say that maybe that's 100 parts per million. If we take and think about all of the parts that might live in that test tube, so all the parts that are water and all the parts that are the red dye molecules, we can say that there are 100 parts of them that are the red circles, the red dye, in the million of total particles. So the million of particles would be all the water particles plus the red dye particles. So this is 100 parts per million particles that are actually the red dye. And what we did when we diluted things is we actually took, remember, this whole thing right here was equal to 10 mils of water. And what we did is we took only 1 mil of it. So that would be like this much. It would be equal to 1 mil of it. We took that 1 milliliter and we put it in here. Now what I did is I was very careful to only take 10 of the dots actually, because 1 milliliter of it is 1/10 of it. And 1/10 of it would be like 1/10 of 100, which is equal to 10. So there are now 10 dots in there. 3, 4, 5, 6, 7, 8, 9, 10. And what we did is we put those 1 milliliter, or 10 parts, into this test tube, and then we filled it up the rest of the way with water. So now these 10 parts live in the whole 10 milliliters of water. So this is now a tenfold dilution. The first dilution is a tenfold dilution. So now we don't have 100%, we are now at 1/10 of it, or 10%. 10% is that number right there. So in other words, what we're looking at is there are only 10 parts per million. So still we have a million parts inside of there, that's all the water, plus the red dye, and only 10 of those million parts is actually the red dye part. And then, once again, we did a serial dilution, which we actually took. If we take 1/10 of this, that would be only 1 milliliter of the 10, and that would only be 1, because there are 10 dots here. So 1/10 of 10 dots is only 1 dot. So then over here, we got 1. My dilution has brought me down to 1%. It is 1% of the original 100% stock solution. And we are now looking at 10 divided by 10 makes 1. 1 part per million. So of all the million particles inside there, we only have 1 part per million. Okay, so let's think about this as a red dye. So now we covered percentage and we covered parts per million. Now I want to talk about the spectrophotometer and how it has the transmittance thing and the absorbance thing. So what you do is you stick these test tubes, or really the cuvettes, which are just a small sample of it, into the colorimeter, so into that box. And into that box we are actually shooting colored light. So there's different colored light. We could shoot red light at it, we shot green light at it, we shot orange light at it, put that in there, and then we also shot blue light at it. And blue light is just at these set wavelengths. That's why you saw like the blue light was at, oh man, I can't remember the number, 450 nanometers. The red light was at 500 nanometers, something like that. So there's the different wavelengths. It just tells you the different colored light. So, let's say we have these pictures right here. So it has all the red dye. If we have red dye, the reason that we see red is because the light that's getting shot at it, the red light, is being transmitted into our eyes. So, if we shot, this right here is all this red here. Let's look at the most concentrated. If I shot red, that's blue. If I shot red light at this test tube right here, what would happen is all of that red light would actually go right through it and be transmitted to the other side. It wouldn't be absorbed, and I would see something like 100% transmittance. I'd see a high transmittance of that. Looking at the next one, if I shot red light through this next one, well, it wouldn't, not as much would go through because it's not as dark of a red color. So a majority of it would go through, but maybe it wouldn't be 100%. Maybe it'd be something like 80% or something like that. Still less. Some of it would be absorbed by the other parts, The other parts, like the water parts, would absorb things, perhaps. And then if you look at the last one, and I shot red light through this one, a lot of it wouldn't get transmitted through. A lot of it would be absorbed because you don't see that dark, rich red color. So maybe this would be at like 20%, something like that. It's still a red color. We still want to be able to transmit red to our eyes, but it's not as deep of a red color, so you wouldn't get as much. Okay, I hope that kind of helps on what the transmittance thing kind of means.